Good morning, everyone. My name is Shen Zhong. I'm from University of California, San Diego. Today, I would like to speak about stress-induced RNA-chromatin interactions that promote endothelial cell dysfunction. Our team hopes to contribute to the spatial mapping of the human cells and tissues by bringing in some molecular interaction assays. In this slide, there are two maps of the same city. The map on the left shows the locations and densities of houses, buildings, land, and water. The map on the right added roads, highways, and ferry lines. In general, a map must include locations and densities of individual objects. In addition, a map can become more informative if the connections of these objects are ended. For example, the map used for urban design can include roads, bike tracks, pedestrian trails, ferry lines, phone lines, electric grids, water pipes, sewage pipes. By analogy, after the spatial distribution of diverse molecular species, such as RNA and proteins, are registered in human molecular maps, perhaps the map can be enhanced by adding molecular interaction information. So the molecular interactions or modifications can include interactions among proteins, RNA, and chromatin. They can also include the addition and removal of chemical groups from chromatin, RNA, and proteins. So our group's interest is in RNA-genome interaction and chromatin-associated RNA. These RNA that are associated with chromatin have emerged as a new frontier of cell type specific regulators of gene expression in humans. Some of the RNA transcribed from a gene can interact with another gene in its genomic sequence in the neighborhood of the original gene, thus forming a cis interaction. And RNA can also travel through a distance to interact with another genomic sequence on a different chromosome, and thus form trans interactions. So these interactions can facilitate the regulation of transcription in multiple ways. For example, RNA can trap transcription factors in the neighborhood of cis regulatory sequence, or RNA can facilitate either the formation or dissolution of condensates near the transcription start sites. So several of the open questions in the field are as follows. First, what are the chromatin-associated RNAs? Meaning, which genes and intergenic regions give rise to chromatin-associated RNAs? Where does each of the chromatin-associated RNA bind in the genome? And thirdly, does chromatin-associated RNA binding correlate with the transcription levels of the target genes? With those questions in mind, through the past several years, several associates in my lab, including Dr. Chui Nguyen and Dr. Wei-Xing Wu, have developed this series of technologies under the name of MARGI, Mapping RNA-genome interaction technologies, to systematically review all the chromatin-associated RNAs and each RNA's respective genomic binding regions. The latest version of these technologies is called iMARGI, in situ mapping of RNA-genome interactions. So the core idea of iMARGI is to first stabilize RNA chromatin interactions by cross-linking, then fragment the genome. After the genome fragmentation, we assume that some of the genomic fragments are devoid of nearby RNA chromatin interactions, which doesn't have an interacting piece of RNA in its spatial neighborhood, whereas some genomic sequences may have attached with RNA molecule. And then we introduce a linker sequence that can be linked on one end to the RNA and on the other end to the fragmented DNA. If indeed there was a piece of RNA that was attached to a piece of genomic segment, then through these two rounds of proximal ligation, we will generate this RNA linker DNA chimeric sequence, and converting this chimeric sequence into double-stranded DNA suitable for DNA sequencing, we can use paired-end sequencing to review both the RNA end and the DNA end of this interaction. So here are a data example. So after mapping a paired-end read to the genome, so we can decipher which part is the RNA end and which side is the DNA end. Here, each row represents a read pair, where the RNA end is denoted in red and the DNA end is denoted in blue. In this particular case, all these read pairs here are approximately 30 to 40 read pairs. Most of them have the RNA end mapped to the malaton1 gene on chromosome 11, and the DNA end mapped to the genomic neighborhood of this KTN1 gene on another chromosome. All these read pairs add together to indicate that the malaton1 RNA interacts with the genomic neighborhood of a different gene on a different chromosome. So one way to visualize this type of genomic interactions in the genome-wide is to use a two-dimensional contact matrix, where here the rows are the linear coordinates of one chromosome, and the columns are the linear coordinates of the same chromosome. Each entry in this two-dimensional matrix represents how much RNA transcribed from the corresponding coordinate on the row to interact with the genomic sequence on the corresponding column. So the higher the red level, the more RNA genomic interaction is happening between those two genomic locations. So here, the higher density on the diagonal line means that the RNA is transcribed from a place and interact with its very nearby genomic sequences. So these diagonal signals represent this majority of interactions in cis, whereas we do observe off-diagonal signals, which represent non-range interactions or interchromosomal interactions. If we were to superimpose the positions of super-enhancers here shown in light blue, we will see that plenty of RNA transcripts are produced from these super-enhancers, and these super-enhancer produced transcripts seem to spread further away from their own transcription loci. So with this type of information, we have started to have some initial answers to the three questions we started with. First, the iMARGI technique reviews hundreds of chromatin-associated RNAs that include link RNAs, small nuclear RNAs, and enhancer-derived RNAs. So these RNAs, I haven't shown the actual data. These RNAs tend to bind transcription star sites in the genome. It turned out that in a given cell type, we typically see more than a third or more than a half of the transcription star sites are occupied by chromatin-associated RNA that were transcribed from other genes, not these transcription star sites gene. And furthermore, we see a global correlation between how much chromatin-associated RNA is bound to the transcription star site to how strongly the gene of this transcription star site is transcribed. So these data and the protocol of the iMARGI technology are described in the listed papers here. So with this information, we started to ask, can chromatin-associated RNA contribute to understand disease etiology? Specifically, we used the diabetic stress to our blood vessels as a model system. We wanted to ask why diabetes-induced blood vessel endothelial dysfunctions seem to be irreversible. There is yet no treatment available to reduce or revert this damage. With this question in mind, Dr. Zhen Chen from City of Hope and our team have teamed up to investigate the endothelial layer of our blood vessel. So endothelium is a single layer of endothelial cells that lines the most inner layer of our blood vessel. That is the interface between our blood and the blood vessel. So Jen's lab has developed an in vitro disease model to mimic the diabetic stress. Particularly, her lab put the endothelial cells under the stress of high glucose and TNF-alpha, which are the typical stress signals in diabetic patients' blood. And under such stress in this time course, from day 0 to day 3 and day 7, the endothelial cells behaved in morphology and molecular phenotypes very similarly to what is observed in patients. And when we used iMARGI to map RNA-DNA interactions in the 3 time points of this time course, we see that in this particular case, the RNA transcribed from a super-enhancer from chromosome 2 didn't seem to interact with any genomic regions of this genomic window on chromosome 7. However, after 3 and 7 days, there starts to form a relatively stable interaction from the super-enhancer derived RNA from chromosome 2 to this target region on chromosome 7. So within the super-enhancer on chromosome 2, there was a relatively understudied link RNA named link 607. And in this target region, there is SUPI1, the SUPI1 gene. This gene is actually a key regulator of inflammatory response. So if we were to plot this 2-dimensional interaction map with the raw read, we see that in control, there were relatively few RNA-DNA interactions between these 2 chromosomal regions, whereas in the stressed condition, we see multiple interactions from link 607 super-enhancer to the SUPI1 genomic region. And we also see mutual interactions, meaning that the SUPI1 RNA also interact with the super-enhancer on chromosome 2. And these data start to suggest interchromosomal interaction that is relatively stable in the disease state, which didn't exist in the control. And to test this, Jen's lab specifically degraded the link 607 RNA in the nucleus. And the nucleus-specific depletion of this source RNA actually reverted the disease-induced activation of the SUPI1 gene. That validates that this disease-specific RNA-chromatin interaction is a cause, and it's a reversible cause of some of the inflammatory-like responses in the endothelial cells. Our next question is whether such RNA-DNA interaction that seems to be at least in part attributable to explain the dysfunctional EC, whether this is a special case or a general phenomenon. To test whether it's a general phenomenon, we reanalyzed the data and analyzed and identified all the potential regulatory targets of this link 607 RNA. That includes our SUPI1 gene, which was shown in the previous slide, as well as a set of other potential target genes, including RUNX1 and TRIO. In order to systematically test all these interactions, we resorted to single-cell sequencing from the control EC and the stressed EC. We also resorted to single-cell sequencing from the healthy donors of ECs and diabetic patients, T2D diabetic, type 2 diabetic patients derived ECs. So our idea is as follows. If indeed this link 607 RNA contribute to activate the expression of a target gene, we would expect that in the activated state, meaning in the disease state, there were more single cells that co-express this source link 607 RNA and the target, in this case, for example, TRIO1 RNA, as compared to the control. So we expect to see a real interaction, a real activation from a source RNA to a target gene to generate an outcome where there are more single cells co-express the source and the target RNA in the same single cell. And this is tested by the odds ratio between the stressed condition single cells and the control condition single cells. The higher the odds ratio is, the more single cells in the stressed condition would co-express the source and the target gene. And we see that nearly the most of the odds ratios are larger than 1, suggesting overall there's a confirmation from the single cell transcriptome. Similarly, in T2D, patients-derived ECs, there are more co-expression of the source and target RNA in the same single cells as compared to the single cells of the healthy donor-derived ECs. Finally, we also see there is indeed quantitative difference between how much response there is in the actual patient's single cell versus the in vitro disease model. That means that not all the actual disease responses are recapitulated in our disease model. Nevertheless, in regard to Lynx 607's response, these two models showed strong correlation. To summarize, why diabetes-induced endothelial dysfunction appears to be reversible. We seem to have seen the following possible causes. First, many RNA-chromatin interactions are formed in diabetic patients' endothelial cells. Second, these chromatin-associated RNA and these RNA-chromatin interactions induce expression of inflammatory genes. Thirdly, degrading chromatin-associated RNA in the nuclei can provide intervention to endothelial dysfunction by keeping the inflammatory genes in a suppressed state. And these data are summarized in the following Nature Communications paper. The people behind the scenes include Ricardo Canandrelli and Wei-Xun Wu, Cui Nguyen, Xiaochen Fan, and Jieju Chen from my lab at UC San Diego. And we have formed a strong collaboration with Dr. Zhen Chen's lab, who has vast endothelial cell experience at City of Hope, especially in Lixia Xu and Yijun Luo in Zhen's lab, as well as Dr. Rama Natarajan at City of Hope collaborated with us for this project. With that, I'm open to any questions. Thank you.